Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. We start the second video, March 24, question paper 1 to question number 22 to 40. Now, coming to question number 22, which statement correctly describes features of stem cells that are essential for their role in cell replacement and tissue repair? Now, when you look at such sort of questions, please read very carefully and try to find out the ones which are totally wrong. And then, of course, you can come to a conclusion, because these are tough questions, and that's why your timing has been increased from 1 hour to 1 hour 15 minutes, because these are now difficult questions. After mitosis of stem cells, the daughter cell can either remain as stem cells or follow a developmental pathway that leads to the formation of specialized cells. No, that's wrong. Is this wrong or is this right? You've got to figure this out. You see, what are stem cells? It says after mitosis, the daughter cells can either remain as stem cells or follow. You still have stem cells in your bone marrow where the red blood cells are being formed. So what we have to understand is that 1 is correct. So if 1 is correct, you've got to understand whether it's wrong, or that's what I said in the beginning, it's wrong, because I wanted to see how many of you would sort of correct me and say, no, Dr. Sarah, you're wrong. It is right. So 1 is correct. Now, why is 2 correct or wrong? We have to see that. Stem cells are different to all the other body cells because they retain all of the genetic information in their DNA throughout the life of the organism. All the cells retain all the genetic material, not just the stem cells. So this is wrong. Then 3, a small population of stem cells is retained in the body of adults throughout their life. Yes, that's what we see in the bone marrow. The cells, the blood cells. Number 4, stem cells have more telomeres. Why would they have more telomeres? Why would they have more telomeres? Telomeres are the same. In fact, telomeres shorten as we age. So 4 is wrong. So that's why the answer is C. I know this was a difficult question. It's not a very easy question, and you all will struggle with this. But that means you read it very carefully. You see which one is right, which one is wrong. And it's not so easy. I'm saying it's a difficult question. I'm not saying it's an easy question. Then coming to question number 23, which statements are correct for all nucleotides? Nucleotides are present where? In DNA and in RNA. Please remember, nucleotides are present in DNA, RNA, and mRNA and tRNA, and all the RNAs, ribosomal RNA. So you read the question carefully. And you see the nitrogen containing base is always attached to carbon atom 1 of the pentose. The phosphate group is always attached to carbon atom 5 of the pentose. A condensation reaction occurs to join the nitrogen containing base. Nucleotides are linked together by condensation reactions between phosphate groups. So the 4 is wrong. This is wrong. They aren't linked together between the phosphate groups. It's between the sugar and the phosphate that the phosphodiester bond is formed. If you knew that, then you would easily be very easy. So that's why the answer is A. 1, 2, 3 are correct. Then we come to another interesting question. How many phosphodiester bonds are present in circular DNA molecule of 2700 base pairs? So this is ATGC. ATGC. So these are 3 base pairs. So there were 2700 base pairs. How many phosphodiester bonds will be formed between them? You see, between this nucleotide and this nucleotide, this one and this one is 1. So between 3 there will be 2. But this then that says is circular DNA. So that means this is joined. That was a little catch. I like this question. So how many phosphodies are present in circular DNA telomeres of 2700 base pairs? So same on this side and same on this side. So that's why it's 5400. But you see, the thing is that you have to understand is ATGC. And this is a circle of ATGC. So that's why there will be 5400 base pairs in the circular DNA. If it was the normal DNA, of course it will be 1 less. Then it will be 2699 plus 2699. How many phosphodiester bonds are present in a circular DNA? So remember, DNA is a double helix. So there is phosphodiester between A and G, and here as well. You have to understand this. Not an easy question. A levels is not easy, it's very tough. Then coming to question number 25, which statements about complementary base pairing are correct? Purines and pyrimidines are different sizes. Yes, one is double ring, one is single ring. Complementary base pairing occurs during translation. What is translation? mRNA to polypeptide. Yes, anticodon, codon. That story is going to take place. The base pairs are of different lengths. No, I'm sorry. That's why DNA double helix. If it is A here, then T here. So base pairs are not of different length, they are same length. That's why it's a parallel DNA. So 3 is wrong. 4, uracil forms 2 hydrogen bonds with adenine. Yes, adenine and thymine. So adenine and uracil will also form 2 hydrogen bonds. So that's why question number 25, the answer is B. 3 was wrong. The base pairs are of different lengths. No. The base pairs are not of different lengths. A and T is always the same length. C and G is always the same length. That's why it's parallel. It's a DNA double helix. Question number 26. During the mitotic cell cycle, the chromosomal DNA is replicated at specific points in DNA molecular replication is occurring are known as replication forks. A typical human chromosome has 150 million base pairs. Of course, for me it was difficult to figure out what is the million. And I had to ring up somebody to tell me how many zeros in a million. I'm sure all of you are better than me at that. It takes about 1 hour. 1 hour means how many minutes? 60 minutes. To replicate the DNA of a typical human chromosome. The rate of replication using a replication fork is approximately 50 bases per second. So how many seconds in an hour? 60 into 60 is equal to 3,600. So 3,600. So approximately how many replication forks must occur? Now I found it very difficult. So 15 million divided by 3,600 and whatever the answer was, and then you divided that by 50. Divide by 50. So 15 million divided by 3,600 and then you divide it and the answer is 835. I know it was difficult. I think this was a difficult question. Question number 27. Which molecule has its synthesis directly controlled by DNA? It's all very simple. I mean, I'm sure you all would have struggled with it because many of you keep on asking me these MCQs and I keep on telling you what is amylase. Then you say, Mrs. an enzyme, and say, what are enzymes? Enzymes are proteins. So DNA controls the production of proteins. This is what you teach in O levels. So DNA controls the production of protein. When I say amylase, you're not correlating protein and the amylase, and you're just looking into it and saying, oh my God, what's the answer? Well, that's all. You're just confused. Question number 28. Which statement correctly describes the association between a companion cell and its sieve tube cell? Now, coming to question number 28, which statement correctly describes the association between a companion cell and its sieve tube cell? The companion cell provides all of the ATP used for energy requiring processes in both cells. Please remember, sieve tube, there is no energy requiring process. It's from high pressure to low pressure, it's mass flow. So A is wrong. The companion cell controls the active transport of molecules through the sieve plate. No, there is no active transports in the molecules through the sieve plate. They just flow by mass flow. The companion cells prevent side-to-side movement of assimilates. Prevent? No, nothing of that sort. So the answer is D. The companion cells provide a nucleus that controls cellular activities in both types of cells. So companion cell has the channel protein, hydrogen ions pumped out, sucrose moves in, sucrose then is through the plasmodesmata, enters the sieve tube. So that is why we are talking about this, and the question and the answer is D. Question number 29. A maize seedling was grown in soil that contained lanthanum oins labeled with a chemical that fluoresces under ultraviolet light. The diagram represents what was observed when a section of root was examined using a light microscope with ultraviolet illumination. Now you can see the fluorescence in the root here is mostly in the cell wall. In the cell wall of the cells, and of course then the Casparian strip is blocked here and they are not going ahead. So then of course it's only the apoplast pathway, it's not the symplast pathway. So then you read all the answers and you come to the conclusion. It's a very simple conclusion. The answer is B. The ions are transported through the apoplast pathway only. Here it says not transported. So this was wrong. Then apoplast and symplast. Symplast was never there. Symplast is through the cytoplasm. We do not see any fluorescence in the cytoplasm. We are only seeing it in the cell wall, and we're seeing it is blocked by the Casparian strip, which is a very, very glaring thing that is blocked by the Casparian strip, which means that it is the apoplast pathway that only gets blocked by the Casparian strip. Question number 30. Which description of adhesion and cohesion is correct? This is a very direct question. I'm going to tell you the answer. Why I'm going to tell you the answer is answer is D, but I'm going to tell you why A, B, and C are wrong. You see, A, adhesion refers to the force between the water molecules due to hydrogen bonding. That is not adhesion. Cohesion is the force between the water molecules. Then adhesion refers to the reduced friction between hydrophobic lignin and the water molecules. Cohesion refers to the forces between and the xylem vessel walls. This is again wrong. Adhesion is to do with the wall. Cohesion is between the water molecules. So this is wrong here again. This is wrong here because of this. Adhesion refers to the reduced friction between the hydrophobic lignin and the water molecules. Cohesion refers to force between water molecules due to hydrogen bonding. So it is adhesion refers to the fact that they stick to the lignin and they help it to move upwards. So it's a matter of knowing your English well and knowing it. So adhesion refers to the force between the water molecules and xylem vessel walls. Cohesion refers to the force between water molecules due to hydrogen bonding. So this was the same in this, but the other part was not correct. Force between the water molecules and the xylem vessel wall. So please, everybody needs to revise adhesion and cohesion because you're bound to get a question on that again. Question number 31. Which conditions are needed to allow the mass flow of sucrose in phloem sieve tubes? So we have the phloem sieve tube in the source and then in the sink. Now in the source, please remember what is going to happen. Sucrose is going to be made. Sucrose moving in, water moving in. More sucrose means lower water potential. Higher hydrostatic pressure and lower water potential. In the sink, what is happening? Sucrose is moving out. So there's going to be a higher water potential and there's going to be a lower hydrostatic pressure. Yes, I agree with it. Sucrose moves out, water follows. But because of the movement of sucrose now, there's going to be less water, so it's going to be more water, less sucrose, more water, so higher water potential. That is why the answer is B. So source and sink, you have to be very clear. What is happening in the source and what is happening in the sink? In the source, there will be higher hydrostatic pressure and lower water potential because sucrose is entering it. In the sink, sucrose is being unloaded. In the source, sucrose is being loaded. So please understand this. Now coming to question number 32, very interesting question. One type of congenital heart defect is where the left and right atria are not completely separated. So here we've got the heart. Here we've got a defect in this, is called the atrial septal defect. ASD usually results in blood moving from the left atrium moving here. So this is oxygenated blood and this is deoxygenated blood. So left atrium into the right atrium. This is the right atrium. And this causes an increased pressure in the right atrium. And right atrium, increased pressure means more increased pressure in the right ventricle, and what arises from this is the pulmonary artery. So the oxygenated blood is entering the right atrium, then the right ventricle, and then it's going into the pulmonary artery. So blood pressure in the pulmonary artery is going to increase. Blood pressure in the aorta is going to decrease because the aorta rises from here. There's less blood here, so less blood in the left ventricle, so less pressure in the aorta. And percentage oxygenation of blood in the pulmonary artery. This is the pulmonary artery which is arising from the right ventricle. So that is going to be increased. Why? Because this oxygenated blood from the left atrium is entering the right atrium and then it is entering the right ventricle and then it's going into the pulmonary artery. So oxygenation of blood in the pulmonary artery will increase. So that is why the answer is D. Question number 33. Which property of water related to its role in blood and tissue fluid is correctly described? Well, water is a solvent for all biological molecules. Protein is a large molecule, is not dissolved in water. Water is a solvent for most non-polar molecules. Wrong. Water requires little energy to increase its temperature because it has a high specific heat. The answer is D. Why? Because blood is required to maintain the temperature. So water cools down slowly because it has a high specific heat capacity. That is one of the role of water in living organisms. Role of blood in living organisms. Blood and tissue fluid, that is going to maintain the temperature. So water cools down slowly because it has a high specific heat capacity. So we enter a cold room, the body temperature doesn't fall down immediately. Question number 34, which statement about tissue fluid formation is correct? The hydrostatic pressure of blood decreases from the arterial end to the venule end of a capillary. Yes, that is correct. So answer is A, but why is B wrong? The water potential is always higher in the blood in the capillaries than in the tissue fluid in the surrounding tissues. Well, no, that's not the case. No cells move out of the blood in the capillaries. Yes, phagocytes do move out. More tissue fluid formation occurs at the venule end. That's totally wrong. More occurs at the arterial end of the capillary. Question number 35. Oxyhemoglobin, carbaminohemoglobin is formed when there's carbon dioxide and it joins. So that will be in the muscle cells or in the body cells. Hemoglobinic acid is when CO2 enters carbaminohemoglobin and bicarbonate ions form. And then the H positive ions connect with hemoglobin and it gives up its oxygen. That also happens in the cells of the body or in the muscle cells. Carbonic hydrase will stay the same all the time. They are found inside red blood cells. How many of these substances will show an overall decrease in concentration as a red blood cell passes through capillaries in the lungs? Now, either you have to know this very well, this chapter, or you just don't know it. So, oxyhemoglobin will increase in the lungs, Carbaminohemoglobin will, what will happen? You see, oxyhemoglobin increases in the lungs, will show an overall decrease in concentration as the red blood cell passes through the capillaries in the lungs. You see, what you have to understand is that what will decrease? The oxyhemoglobin will increase, the carbaminohemoglobin will decrease, and the hemoglobinic acid will decrease. So the answer is 2. Carbaminohemoglobin will give up its carbon dioxide. So if they were 100, they're going to become 0. The hemoglobinic acid will be broken up because the hemoglobin now is not friendly with the hydrogen, now it's friendly with the oxygen. The hemoglobin is sometimes friendly with the oxygen, sometimes forms oxyhemoglobin, sometimes friendly with the H ions and forms hemoglobinic acid. So, but when it's at the lungs, it forgets its friendship with the H ions and then becomes friendly with the oxygen. So the hemoglobinic acid gives up its hydrogen. So the hemoglobin is flirting around with oxygen and hydrogen. Sometimes it's friendly with the hydrogen, sometimes it's friendly with the oxygen. So having a good party time. So the answer was B. 2 of these will decrease. Then it's question number 36. Which of these structures typically contain cartilage and cilia? Bronchi, bronchioles, and trachea. Cartilage and cilia. Bronchi, bronchioles, and trachea. C, 1 and 3. Bronchi and trachea. You see, bronchioles do not have any cartilage in them. Cilia will also not be present. So 1 and 3 only. Then question number 37. Which statements about the function of tissues found in the human gas exchange system are correct? Collagen in the bronchi prevent them collapsing. Smooth muscle in the bronchioles can contract to increase the flow of air. Contract, so that means if they contract, they become smaller lumen. Elastic fibers in the alveoli stretch and recoil during breathing. So collagen in the bronchi prevent from collapsing. Smooth muscle in the bronchioles can contract to increase the flow of air into the alveoli. That's totally wrong. Elastic fibers stretch and recoil. But the question was, which statement was the function of tissues found in the human gas exchange? So it's not collagen, it's cartilage in the bronchi prevent it from collapsing. Cartilage in the bronchi prevent it from collapsing. 3 is only correct. So that's why the answer is D. Collagen is not present. Collagen is present, but it doesn't prevent it from collapsing. It's the cartilage. You see, they use the word C collagen and C cartilage to confuse you and me. I got confused as well. So 37, the answer is D. Question number 38. Antibiotic resistant strains of Mycobacterium tuberculosis are a major problem when treating TB. A new antibiotic, Teixobactin, could be very effective at killing M. tuberculosis with only a small risk that the bacteria will evolve Teixobactin resistance. When does resistance occur? When there is a mutation in the plasmids. Penicillin and similar antibiotics bind to a single protein, but Teixobactin binds to two lipids that are needed for the formation of the bacterial cell wall. So this is how the antibiotic works. Teixobactin binds to regions of the two lipids that do not vary across many different species of bacteria. Which statement helps to explain why the use of Teixobactin is thought to be less likely to lead to the evolution of antibiotic resistance than the use of many other antibiotics such as penicillin? This question is a typical test of how good your English is. A single mutation can result in bacteria that are resistant to penicillin and similar antibiotics, but at least 2 mutations are required to produce Teixobactin-resistant bacteria. Fine. Number 2, mutations can affect the structure of proteins but cannot affect the structure of lipids. Now, this does not give us an explanation of this. It says which statements help to explain. That's why I said it's just English, it's not even bio. The lack of variation across many species of bacteria in the two lipids that bind to Teixobactin suggests that the particular structure of these lipids is essential for successful bacterial cell wall formation. Well, that's fine. So that is why the answer to 38 is C. 1 and 3 only give a statements which help to explain why it is giving resistance. Then coming to question number 39, monoclonal antibodies are now being used to treat some human diseases. What explains why monoclonal antibodies are suitable for this purpose? They can divide by mitosis. Who can divide by mitosis? Not monoclonal antibodies cannot divide by mitosis. They are just antibodies. So this is wrong. It is the plasma B cells that divide or the hybridoma cells that divide and produce the antibodies. They are specific to a particular antigen. They can be modified so they do not act as. So we're giving the monoclonal antibodies as a treatment. Remember that. We're doing that in cancers as well. So they can be modified, they do not act as antigens themselves. That is why the answer is C. 2 and 3 only are correct. That is why they are being used to treat human diseases. They're being used in many cancers. So this is how you have to read it carefully and answer it. Last question. Measles is an infectious disease caused by a virus. The graph shows the number of cases of measles each year in a country before and after a vaccine was introduced. So vaccination was introduced in 1964-5, somewhere around that period here. And then of course we see a decrease. What could have caused the decrease in the number of cases of measles after vaccination was introduced? Keep on looking at the graph and then come up with the answers. So vaccine provided artificial active immunity. What is it? What is in the vaccine? The vaccine is a weakened form of the pathogen. Vaccine is a weakened form of the pathogen. So yes, it does provide artificial active immunity. It does not provide artificial passive immunity. Artificial passive immunity is when you give tetanus ready-made antibodies. So this is also correct. It does not provide artificial passive immunity. Fewer people are able to act as hosts for the virus. You see, if the number of people suffering from it decreases, so there will be lesser number of people who can have the virus and then spread the virus. So yes, fewer people act as hosts for the virus. So that is why the answer to this is A. Thank you very much and all the very best.